We'll next hear from Branden Van Oss from Karen Arndt's lab, who's going to tell us about what they've learned recently about Paf1.
Thanks, Lorraine. Thank you all for being here today. And I'm excited to have the opportunity to tell you about some recent progress that we've made as part of a collaboration I've been a part of in understanding what's really been a long-standing question in the Arndt lab, and namely how a conserved transcription elongation factor works to promote an important post-translational histone modification. So in the Arndt lab, we study transcription in yeast, and much of our work focuses on the 5-subunit yeast Paf1 complex, or polymerase-associated factor 1 complex. And as the name implies, the complex directly associates with RNA polymerase II, and it regulates eukaryotic transcription in a number of ways. This includes regulating the phosphorylation status of the RNA polymerase C-terminal domain, or CTD, maintaining proper nucleosome occupancy in the wake of a transcribing polymerase, and promoting proper termination in 3' end formation. But the function that is most important to the story I'm going to tell you today is that it's required for the establishment of several post-translational histone modifications. And one modification that we're particularly interested in is the monoubiquitylation of histone H2B at a specific lysine residue, lysine 123 in yeast, or the orthologous K120 residue in humans. And it's important to note that as far as we know, the Paf complex has no enzymatic activity, and so in the cell, deposition of this mark is carried out by the E2 and E3 enzymes, Rad6 and Bre1. And H2B ubiquitylation sits at the top of a cascade of histone modifications, namely methylation events on histone H3, some of which we've heard about today, specifically H3K4 and K79 methylation by the set1 and dot1 enzymes, respectively. And we're interested in H2B ubiquitylation because it plays a number of important regulatory roles, not only in yeast, but also in higher organisms. It regulates the transcription initiation and early elongation steps. And although the mark is associated with active transcription, it's also important for proper silencing of heterochromatic regions, such as telomeres. It's recently been shown to regulate release from pausing in metazoans, and in higher eukaryotes, it has roles in several fundamental processes, including differentiation, development, and when misregulated, can lead to cancer. So one of the first questions we can ask is, what are the contributions of the individual subunits of the Paf complex in establishing normal levels of H2B ubiquitylation in the cell? So here I'm just showing a Western blot probing for ubiquitylation in strains where we've deleted individual subunits of the Paf complex. And you can see that while several subunits have a role in establishing normal levels of the modification, only the Rtf1 subunit is strictly required for detection of this mark in vivo. You can compare this to a control strain in which the site of the modification has been mutated to an arginine. Now, previous work in our lab through deletion analysis had identified a small region of Rtf1 required for H2B ubiquitylation and its associated modifications. And what I'm showing here is that if we overexpress this region, which we term the Histone Modification Domain, or HMD, we can restore wild-type levels of H2B ubiquitylation in an Rtf1 deletion strain. Through a genetic screen, we also identified a substitution within the HMD, the E104K substitution, which completely abolishes HMD activity. So this was really cool, but since we know that other Paf subunits have a role in promoting H2B ubiquitylation, we wanted to ask what would happen if we overexpress the HMD in a background where all 5 Paf complex subunits have been deleted. And so that's what I'm showing here. And you can see that when we do so, we're able to restore robust H2B ubiquitylation levels even when expressing a minimal HMD fragment consisting of as little as 66 amino acids. And that leads me to our central question that we've been exploring, which is, what is the mechanism by which the Rtf1 Histone Modification Domain acts to promote H2B ubiquitylation? And to begin to address that, we first undertook structural and genetic analysis of this domain. So what I'm showing here is the crystal structure of the HMD, just this 66 residue fragment of Rtf1, solved by our collaborators in the Van de Mark lab. And the residues that I'm showing are all residues that were subjected to mutagenic analysis, in most cases substitution with an alanine. And those residues that are indicated in yellow are those where the mutant proteins exhibited a significant defect in H2B ubiquitylation. And what you can see is that many of these functionally important residues cluster along a ridge on the large helix of the HMD, helix B. Now, having identified a functionally important surface on the HMD, we hypothesized that perhaps the HMD was promoting H2B ubiquitylation through a direct interaction with another protein. And given the rapid kinetics of the ubiquitylation reaction, that this interaction might be transient. And so in order to test this, we undertook a site-specific in vivo cross-linking approach. This approach makes use of a non-natural photoreactive phenylalanine analog known as BPA. And in this system, we overexpress Rtf1 derivatives in which a particular amino acid within the HMD has been mutated to the amber codon. And an engineered tRNA and tRNA synthetase will recognize this amber codon and incorporate BPA into the elongating polypeptide. And upon exposure to UV, we induce cross-links between Rtf1 and any interacting proteins. And what we were excited to find by this method was a direct interaction between Rtf1 through the HMD with the E2 enzyme that I told you specifically catalyzes this mark in vivo, Rad6. So what I'm showing here is a Western blot probing for MYC in a strain in which Rad6 is MYC tagged, in which we're overexpressing Rtf1 derivatives that have BPA incorporated at the specific sites indicated at the top of the blot within the HMD. And what you can see is that for 3 of these BPA-containing derivatives, specifically when we incorporate BPA at residues 105, 108, or 112, we see a super-shifted band that's BPA-dependent, indicating a direct interaction between Rtf1 and Rad6 through the HMD. And importantly, when we make the inactivating E104K substitution in some of these same derivatives, we see a complete loss of this band, indicating a loss of an interaction and indicating that this interaction is functionally relevant. And when we map to the surface of the HMD regions where we did and did not detect Rad6 cross-linking, we see that the region where we detect cross-linking clusters to a surface-exposed region directly adjacent to the critical E104 residue. Now, we also tested another surface of the HMD that contains several highly conserved residues and did not detect Rad6 cross-linking there, perhaps speaking to an alternative or a separate function of the HMD. So I'm going to switch gears a little bit here and ask another question, which is, what role, if any, do other Paf1C subunits play in regulating H2B ubiquitylation? And to address this, we initiated a collaboration with Dr. Frank Pugh's lab at Penn State. And they've developed a variant of the chromatin immunoprecipitation, or CHIP technique, known as ChIP-EXO, which allows us to map the genomic localization of chromatin-associated factors at extraordinarily high, nearly single base pair resolution. So what I'm showing here is ChIP-EXO data for all 5 subunits of the Paf complex. The gray shading indicates histone H2B, and with the resolution of this technique, we can actually distinguish the individual H2B molecules within a particular nucleosome, which are the peaks that you see. The x-axis is centered around the +1 nucleosomal dyad, the first nucleosome after the transcription start site, and the y-axis indicates the relative occupancy of these proteins. You can see that all 5 subunits co-localize as you might expect for a stably associated complex, and their enrichment is greatest in the 3' end of the gene, particularly around the +3 and +4 nucleosomes. So what do we see when we look at the H2B ubiquitylation mark itself? So here I'm showing a similar graph. Total H2B, relative levels of total H2B are shown in red, and relative levels of the modification are shown in gold. And you can see that similar to the Paf complex itself, enrichment of H2B ubiquitylation is greatest in the 3' region of the gene, particularly at the +3 and +4 nucleosomes. We next wanted to ask what H2B ubiquitylation would look like if we expressed just the HMD in an Rtf1 deletion background. So here H2B is shown in gray, and the solid gold line represents our control where we put back full-length Rtf1, and we see, as with the endogenous protein, that enrichment is greatest at the +3 and +4 nucleosomes. However, when we express the HMD in an Rtf1 deletion background, which we know from previous work means that it's unable to associate with the other members of the complex, we observe a 5' shift in the enrichment of H2B ubiquitylation. In particular, we see greater enrichment near the +1 nucleosome, which tells us that even though we know that by Western blot the HMD is capable of promoting wild-type levels of this modification, the other members of the complex are important for targeting the modification to its appropriate genomic location. Now, finally, it's been suggested for some time that perhaps the Paf complex promotes H2B ubiquitylation through a simple recruitment model by recruiting Rad6 and Bre1 to the site of the modification. But the fact that we saw enrichment at the precise locations in the genome that the Paf complex was present led us to ask whether the Paf complex through the HMD might be stimulating H2B ubiquitylation more directly. And in order to test this, we made use of recombinant factors and a minimal in vitro ubiquitylation assay. This assay just contains the minimum components necessary for H2B ubiquitylation in vitro, namely the E1, E2, E3 enzymes, a source of recombinant nucleosomes, and ubiquitin and ATP. And what I'm showing here is a time course, and you can see that even without the HMD or a source of Rtf1, we're able to achieve robust H2B ubiquitylation after 30 minutes. However, we see that when we add the HMD to the reaction, we see about a 4-fold stimulation in the amount of H2B ubiquitylation. And we also see that when we make that E104K substitution in the HMD, any stimulatory activity is completely lost. Finally, one experiment that I don't have time to describe in detail today, we found that when we carry out the reaction without the E3 enzyme, Bre1, we lose any stimulatory effect of the HMD. This led us to conclude that the HMD, rather than directly stimulating Rad6 catalysis per se, might be helping to direct it to its appropriate target lysine on H2B. So in summary, I've shown you today that overexpression of the HMD restores normal levels of H2B ubiquitylation even when all members of the Paf complex are absent. I've shown that Rtf1 directly interacts with the E2 enzyme that catalyzes the modification through a conserved surface on its histone modification domain, and that this interaction is dependent on the ability of Rtf1 to promote H2B ubiquitylation. I've further shown that the Paf complex functions to regulate H2B ubiquitylation patterns by targeting the HMD to its proper genomic locations, and that the HMD acts as a cofactor in the ubiquitylation reaction by directly stimulating Bre1-dependent H2B ubiquitylation. There are a lot of people to acknowledge on this project. Members of the ARNT lab, those shown in bold, have contributed directly to this project, as well as our collaborators in the Vandemark lab and in the Pew lab. And I'd also like to thank Drs. Jae-Hoon Kim and Song Tam for providing reagents and our sources of funding. And with that, I'd be happy to take any questions you might have.
Maybe as chair's prerogative, I'll start with one. In those really lovely surface mapping studies, you raised the idea that there might be a second function for the HMD that's not Rad6 bound. Would you like to speculate about a model for that?
We really don't know what that other function might be. There are other phenotypes that are associated with the HMD. Mutations in the HMD can lead to a loss of telomeric silencing and cryptic initiation phenotypes, but those phenotypes, at least the telomeric silencing phenotype, seems to be associated with the ubiquitylation function. But that surface contains several very highly conserved, in some cases invariant residues, which makes us think they must be important, but we really don't have a clue yet as to what that function might be.
I have a question about your chip exo.
Yes.
Have you considered analyzing the data and taking into consideration the expression level of the genes, and if the Paf1 distribution might change?
Yeah, we did look at protein levels of all the factors that we chipped. Which factors are you asking?
No, I mean the targets. So you did whole genome distribution
Yes.
of the factors. If you took into consideration the expression of those genes, would you anticipate the ubiquitin mark and the Paf1 localization to change from the 3' end?
So you're talking, if we take into account expression of the Paf complex subunits?
Not the complex, the actual genes
Oh,
that you're chipping.
the genes at which it localizes. Yes. Okay, so we haven't, so yes, we did look at that. And it's been known for a long time that the Paf complex is enriched at highly transcribed genes. And so is H2B ubiquitylation. And it's also thought that transcription itself is a prerequisite for ubiquitylation. So you do see higher levels of both Paf complex members and H2B ubiquitylation at highly expressed genes. Sorry if I misunderstood the question.
Final question.
Yes, so you may have shown this, but does the HMD domain bind to chromatin? Is it?
It does. So we have chip exo data that I can show a slide if we can do that.
It's okay, but is it also
yes,
more
it does, and unlike the wild type protein, its association with chromatin is dependent on its ability to promote ubiquitylation. And so the Paf complex normally is tethered to chromatin by interactions with the polymerase as well as SBT45. But the HMD's ability to associate with chromatin, we think, is dependent on its ability to interact with Rad6, because when we make that E104K substitution, it can no longer associate with chromatin.
And so maybe a quick question. And so
Sure.
I thought that, if I remember correctly, Bre1 actually can recruit Rad6 to chromatin, right? Is that correct?
Yes.
And so now you show that the HMD actually binds to Rad6, but so how should I see the order of recruitment?
So we did look at what role Rtf1 might be playing in Rad6 and Bre1 recruitment. And yes, Bre1 does recruit Rad6 to chromatin, but what we found was that when we delete Rtf1, we see a reduction in Bre1 and Rad6 occupancy in the gene body, particularly at highly expressed genes. But that's not enough to necessarily explain the fact that we have a complete loss of the mark in an Rtf1 deletion strain, which makes us think it might be doing something a little bit more than just recruitment.
Thanks again, Branden.
Thank you.